least I caught it. Okay, so we're recording. I forgot to hit record, so if you're watching this on the recording, I've already gone over the first question. But if you're watching this from the recording, you can check out my session from either Sunday or Wednesday of this week because it's the same content. And I will not forget to record on, well, I recorded last night and I'll make sure to remember to record on Wednesday. So, okay, sorry about that. So getting into primary endosymbiosis theory. What is it and what pieces of evidence exist for this theory in the context of the mitochondria and in chloroplasts? So kind of where this theory came about is researchers discovered that two organelles within eukaryotes have their own separate DNA. So a eukaryotic cell has its own DNA, obviously, but then it was discovered that each of these organelles also contain DNA that is different from the DNA of the eukaryotic cell. So that kind of raised a red flag. I mean, something was kind of like off there. Maybe not off, I guess I shouldn't say that, but it was definitely like interesting. Also, I should point out, I want to say every eukaryotic cell has a mitochondria. There might be some very few and far in between exceptions. I'm always very hesitant to say like, oh, everything fits under this umbrella, because in biology there do tend to be a lot of exceptions. But for the most part, you can think of every eukaryotic cell having a mitochondria, or having not just one singular mitochondria, but they have mitochondria throughout their cell. But chloroplasts are not found in all eukaryotic cells. So animal cells, for example, don't have chloroplasts. They are the organelle in which photosynthesis takes place. We know animals can't do photosynthesis, so that makes sense. So chloroplasts are more associated with plants, algae, that sort of thing. Okay. So what is primary endosymbiosis theory, or endosymbiont theory, whatever you want to call it? Basically, what it says is it's proposed that an early eukaryote engulfed an aerobic, well, yeah. Let's go with that. Maybe we'll take that descriptor out and we'll just say prokaryote to be more general, and established a symbiotic relationship rather than digesting. Yeah, I hope no one saw me using the wrong then right there. But I fixed it. So yeah, basically kind of the theory is that they're back in the day, not like 100 years ago, like way back in the day. I don't actually have a number off the top of my head of how long ago it was, but very long time ago. Basically what researchers have come to agree on is that there were, eukaryotes, prokaryotes kind of coexisting, and that an early eukaryote engulfed one of the prokaryotes that it was nearby. And that engulfment, I guess you could say, was probably like, typically I think that would be associated with, engulfing a smaller prokaryote for nutritional value, for consumption, to eat. But instead of that eukaryote eating the prokaryote, it established a symbiotic relationship with it. So you don't really need to worry about the nitty-gritty behind that of like, how did it actively choose, I'm not going to eat this, I'm going to develop a relationship with it? Probably not. But you don't need to worry too much about the how that happened. Yeah, but basically, so they developed this relationship that was beneficial to the eukaryote because some prokaryotes have abilities that eukaryotic cells don't have. Sorry, for the distraction, I had a marker in my hand and I realized I've just been scribbling all over my ankle. I thought the cap was on it, but it's not. Anyway, so sorry about that. I need to get it together, I think. I'm a little bit all over the place lately with my PAL sessions. Anyway, okay, so back to this. Where was I? The early eukaryote benefited from establishing this symbiotic relationship with the prokaryote because some prokaryotes have abilities that eukaryotes don't have. For example, some prokaryotes are able to okay, I don't actually know. I don't want to get into like the super specifics of it. So I'm trying to think of how much I should, because I can't remember. Okay, scratch the last 30 seconds. Let's just say that by engulfing these prokaryotes, the eukaryotes developed abilities through these prokaryotes that they couldn't have done without them. So without developing, or without engulfing the prokaryote that turned into the mitochondria, that early eukaryote wouldn't have been able to produce ATP in, the mitochondrial electron transport chain that we learned about in the previous course, without engulfing the prokaryote that ended up evolving into a chloroplast, that early eukaryote wouldn't have been able to do photosynthesis. So yeah, okay, I think I was going to explain that in a confusing manner, but I'm not going to do that. You're probably already confused because I'm a little bit incoherent this evening. But moving on. If there are any questions, let me know, as always. But anyway, so what pieces of evidence exist for this theory in the context of mitochondria and in chloroplasts? The pieces of evidence are kind of similar, actually. But essentially, for the mitochondria, evidence includes that the mitochondria exhibits two membranes. One from the host and one from the bacterial endosymbiont, which is the bacteria that formed the inside symbiotic relationship. So if you remember looking at kind of the structure of the mitochondria when we talked about ATP production, cellular respiration, that's the word I'm looking for. It has that outer membrane, and then it has the really wonky kind of like squiggly inner membrane. And that's pretty unique. It's not entirely unique to the mitochondria, as you'll see in a second, but it's pretty unique to other membrane-bound organelles, such as the nucleus. The nucleus has one membrane, not two. So the other big piece of evidence for the mitochondria evolving this way is that I mentioned earlier that mitochondrial DNA exists. So DNA that is unique to the mitochondria, and that DNA, the chromosomes within that DNA, are circular. So we talked about prokaryotes last week, or maybe the week before. Anyway, lecture 9, I think, whenever that was. And we said that prokaryotes have this distinctive circular chromosomes. So that definitely got people thinking, because it's not at all typical for eukaryotes to have circular chromosomes. And also, after like some analyses on the DNA within the mitochondria, it was discovered that the DNA of the mitochondria is closely related to a specific type of bacteria called alpha proteobacteria. So yeah, they're more closely related to this type of bacteria than they are to eukaryotic bacteria, or they're more closely related to this type of DNA than they are to eukaryotic DNA. So I think that says a lot in that. I don't think, that the DNA in the mitochondria came from the eukaryotic DNA. I think it came from some prokaryotic DNA, likely because it was engulfed. It initially existed as a prokaryote. That prokaryote was ingested. And it survived and it evolved, and now it is this remarkable organelle that is the powerhouse of the cell, as we all know. Okay, so that's the mitochondria. Chloroplasts are really, really similar evidence-wise. They also have two membranes. Whoops. So if you remember looking at photosynthesis and the light and dark reactions, and if you remember kind of taking a look at things taking place within the chloroplast, there are two membranes, kind of the outer, and then there's like an inner that kind of, well, I don't think I need to explain what it means to have two membranes. Again, I overcomplicate things. Anyway, okay. So similar to the mitochondria, the chromosomes of chloroplasts are also circular. And these chromosomes are closely related to cyanobacterial DNA. So again, another type of bacteria that have really, really similar DNA to the DNA found in chloroplasts. So again, likely, this provides more evidence to the theory that this is how the chloroplast came to be. It was likely a cyanobacteria, I guess, just living its best life as a free cell, and then it was engulfed and it survived, it wasn't digested, and it established this relationship. And then it evolved, mechanisms became a little bit more complex, but again, we don't have to worry about how that all happened. Okay, I think that's all I want to say for that. Personally, I found when I took this course that this was a little bit confusing to me. I think generally kind of my peers and classmates felt the same way. So if you're finding this confusing, don't be like bogged down or concerned. Because personally, like in my experience and knowing like how people felt about it when I took the course and how I did Pell last year, people do find this confusing. So it's kind of one of those things, in my opinion, that you're not really going to pick up the very first time you hear it. So you might need to like rewatch. Well, there was the Amoeba Sisters video posted on D2L. I'm a big video fan. I find videos with like a graphics and that sort of thing very helpful. But anyway, yeah. Personally, I find it's one of those things you don't really pick up right away. I mean, maybe some people do. Everyone learns differently, so. Okay. Yep, I think that's all I wanted to say for that. So, moving along into lecture 11 and talking about something that we've kind of talked about a lot at this point, if you include kind of the precursor course, or the prerequisite, I guess is the better word, being Dr. Patton and Dr. Durnford's lecture. What are the strengths and limitations of asexual and sexual reproduction? Yeah, we've talked about this a lot. You know, there could be people taking this course though that I think the first course is like a prereq, but you might have taken it last year or the year before. So it's good to go over this too because it is important. So starting with asexual reproduction, some of the strengths, there's really one kind of, well, no, I shouldn't say there's just one. A big strength is that there is greater reproductive output in asexual reproduction. So basically what this means is asexual or organisms that reproduce asexually tend to have a lot more offspring. It doesn't necessarily mean that they're going to have a lot more offspring that survive and reproduce. Sometimes asexually reproducing organisms just leave a ton of offspring and they don't really do much to kind of like protect their offspring, so a bunch of them die off and it ends up that they only have like a few that survive, but they still have a few that survived and they really didn't have to do any work in protecting them, that sort of thing. So yeah, they have a greater reproductive output generally when compared to sexually reproducing organisms. Asexual reproduction is also good in a stable environment. Kind of like if it's not broke, don't fix it. Essentially, there's not going to be a lot of changes in asexual reproduction because we know asexual reproduction is one parent and their offspring are genetic clones of that parent. So if the parent is really, really suited to the environment that it's in, and the environment's not changing, then why would you want genetic variation in the offspring? You know, you would just want the offspring to be the exact same because that worked out for the parent really well. It's also good in species with rapid generation times. And that is because we see increased mutation when generation times are rapid. So bacteria, for example, reproduce asexually through binary fission, typically. And there are, you know, some species of bacteria can double their generation in like 20 minutes. Others, you know, take longer, but that's pretty remarkable that every 20 minutes you can double the population. So you can start it with one bacterial cell, in 20 minutes you have 2, in 40 minutes you'll have 4, in an hour you'll have 8, and so on and so on and so on. Okay, so with, okay, I'm missing a word here. Good in species with, I'm missing several words here, with rapid generation times, sorry. So with rapid generation times, you are reproducing over and over and over again, really quickly. And so with all of these rounds of reproduction, you're going to see more mutation simply due to the fact that you're reproducing more. And every time that reproduction occurs, there's a chance of mutation. So if you're pumping out a ton of offspring all the time, then there's just an increased chance that there are going to be mutations involved. Yeah, and I think a lot of times mutation kind of gets this negative connotation that it's a bad thing, but it's really not. Sometimes mutation can be really beneficial. Sometimes mutations can be detrimental, but not typically. Well, I shouldn't say that. It really depends on the situation. But they're not always bad. Sometimes they're really beneficial. Sometimes they're neutral. They don't really do anything. Anyway. So that those are the strengths of asexual reproduction. But one of the limitations of asexual reproduction is there's no recombination. And that's not ideal in certain environments because we know recombination is really important for evolution. So if you're, in an unstable environment, for example, and you lack this ability to get a lot of genetic variation, that's going to mean you're lacking an ability to, I don't want to say you're lacking the ability to evolve because it's still possible, but recombination generates new genotypes, and these new genotypes facilitate natural selection, and that allows for evolution to act on populations. Okay, so comparing that to sexual reproduction, obviously one of the big strengths, kind of the opposite of what I just said, recombination, which generates new genotypes, and that facilitates natural selection. These. And it also allows for longer generation times. So if you think of any organism that reproduces sexually, let's go with elephants. I know they have, I don't know the number off the top of my head, but they have a really long gestational period. Humans are 9 months. I know that one off the top of my head, obviously. Elephants are even longer than that. They have, a very long generation time. They're definitely not, and not even, I guess, just that it takes a long time to, for an elephant to grow inside of the parent. When that elephant is born, it's not going to immediately reproduce either, A lot of sexually reproducing organisms don't reproduce right away, like a bacteria would, for example, or, other asexually reproducing organisms. One of the limitations of sexual reproduction would be reduced reproductive output. And again, that's in comparison to asexual reproduction. They just can't create as many offspring as fast as asexual reproduction can. Okay, sorry, I wrote something down wrong on my answer sheet, so I was looking at that like, what am I trying to say? Anyway, so yeah, they just simply can't reproduce at as high quantities as an asexually reproducing organism could. But typically, a rule of thumb is that in asexual reproduction, you're going to have quality over quantity. So like I said earlier, think of some asexually reproducing organisms just kind of throw all of their eggs in one basket. I don't know if that's the right phrase, so let's not use that. But anyway, kind of what I think of when I think of this is if you've ever seen one of the house plants that are called, I've heard them called mother of thousands, mother of millions, that sort of thing. But essentially they're this house plant and they have these long leaves, and certain times of the year they get these little tiny little seedlings on all over the plant, on the leaves. And there's just tons of them and they fall off at the slightest touch. And if you were to take one and bury it in soil, it'll grow up into a plant that is a genetic clone of the parent from which it came off of. So that would be an example of asexual reproduction being, there's a great reproductive output because there's tons of these little seeds. That's why they're called mother of thousands or mother of millions, because there's so many of them. But are all of them going to fall off into the nearby soil and start growing? I mean, maybe, but maybe not also. Um, Highly unlikely that every single one is going to drop off into the soil and grow up into another plant. It's just not feasible that all of them will, but some of them will. So that's an effective strategy in ensuring that you have some offspring without putting a lot of energy into kind of looking after your offspring. Whereas if you think of mammals, for example, with sexual reproduction, typically, I mean, it depends. If you think of like a coyote or wolf, they'll probably have, I don't know off the top of my head, but probably like a dozen maybe puppies in a litter. So that's kind of a lot. But if you think of maybe like a bigger mammal, like a deer, for example, I'm kind of in a rural area. I see a lot typically of baby deer in the spring. Not a lot, but quite a few. Usually a white-tailed deer will have one or two, sometimes three. I think three is pretty much pushing it though. And actually, that's a good example. But they have to exert a lot of energy into looking after their offspring. So they have a lot less offspring. So they really have to make sure that they're putting a lot of effort into their offspring so that the offspring survive, eventually reproduce, pass their genes on to the next generation's gene pool. So sometimes I said, oh, that was a good example because I was thinking about deer. Typically, it's really sad, but sometimes you'll see, I'm going to get emotional talking about this. I get very attached to the deer in my backyard, but sometimes you'll see in the springtime, deer, and she'll have two babies, and then all of a sudden, just one day, she's only got one. And it's really sad, it's part of life though. But with one, either maybe it's been deserted because the mother couldn't take care of it and she knew she had to put all of her effort into the other one in order for it to survive, that sort of thing. It's really complicated. We don't really need to get into it. But that's kind of an example of how, I guess, sexual reproduction favors quality over quantity. Asexual reproduction favors quantity over quality. It's probably a side tangent. It's probably not crucial for a midterm or anything, but I think it's important as a concept. Okay. So actually, maybe I'll put this right on the next page so we have lots of space with that diagram. The 3 sexual life history patterns. So these are all related to sexual reproduction, but it's not just like there's one type of sexual reproduction, this is how it's done, and that's it. We kind of classify it into these 3 different patterns, with diplontic on one end, haplontic on the other, and haplodiplontic kind of meeting in the middle, but really it's more of a continuum. But anyway. Okay. I kind of took up a lot of time already, but I think we still have lots of time. 20 minutes. So diplontic, we'll start there. Diplontic is probably the easiest to understand in my opinion because animals are diplontic. So that means humans are diplontic and we understand that pretty well. So in diplontic life history patterns, gametes are haploid, but all other cells are diploid. So if we're going to talk about haploid and diploid, I should probably kind of address what that means. Essentially, we as humans are diploid, as this implies. Our gametes are haploid. What does diploid mean? Think of the prefix di, which often means 2. We have 2 sets of chromosomes. So humans have 46 chromosomes, but they're in pairs. So we have 23 pairs of chromosomes. So we are diploid because our chromosomes are in pairs. But our gametes, so sperm cells, egg cells, are haploid. They only have 23 chromosomes. They just have the one set. So essentially, see in this, I really like this diagram actually, I find it helpful. Anything up here in the green area is where the cells are going to be haploid. Anywhere in this red area is where the cells are going to be diploid. So we'll start here. We have gametes, so sperm and egg. Fertilization occurs, and when fertilization occurs, that is when we go from being N to 2N. So we have two cells that are haploid, each with 23 chromosomes. They come together. Now we have 46 total, and they're going to be, 23 pairs of 2. So mitosis occurs, cells proliferate, and eventually we end up with this diploid multicellular organism. And yeah, like I said, all animals are diplontic, so that could be a variety of different things. But so then when this diploid multicellular organism is ready to reproduce, it'll undergo meiosis, which we know is the process of production of gametes. And so the gametes that are produced will be haploid also. And the cycle continues. A point about diplontic, I'll just write that in there. Animals, not only, including humans, undergo this diplontic cycle. And I'll go with haplontic next. So haplontic is kind of the opposite of diplontic. Most cells are haploid, with the exception of diploid zygotes. So the only time that haplontic organisms are diploid is just after fertilization. So essentially what happens is we have haploid gametes, similar to over here. They come together, fertilization occurs, we have a diploid zygote. So we go from 23 to 46, but not actually because humans don't do this. But we go from N to 2N, doubles. And we have this diploid zygote. And so unlike over here, mitosis isn't going to occur. You're not going to get a multicellular diploid organism in haplontic, but meiosis will occur. Spores will be produced, i.e. gametes, and these can go on to grow into either single cell or multicellular organisms. But the organism is going to be haploid. So only the zygote is diploid. And I guess the diagram says it, but I'll write it in. Most fungi do this, as well as some plants. Or not some plants, sorry. Some algae. I guess it says it right there, so you knew what I was going to say anyway. Okay, probably the more confusing one is haplodiplontic. It can be intimidating, I guess, because it looks confusing, but it's kind of just a combination of the two. And kind of the key phrase with haplodiplontic is alternation of generations. And what that means is from generation to generation, we're alternating between haploid and diploid. Haploid, diploid. So these types of organisms can exist as either. So if you look at the diagram, we'll start here because that's where we've been starting. We have these haploid gametes, just the same as we've seen everywhere else so far. Fertilization occurs. After fertilization, the cells are diploid. Here, mitosis can occur. We can get a multicellular diploid organism, just like we saw over here in diplontic. And then same thing, it'll go through meiosis. Gametes will be formed, but this is where it differs from diplontic, and the haplontic stuff comes in, is that those gametes can grow and or not grow, I guess, proliferate, and you can end up with a haploid multicellular organism. And then when, basically, yeah, it just follows a cycle. So it's very much a combination of these two. It can exist as diploid, undergo meiosis, exist as haploid, exist as diploid, meiosis, haploid, alternating kind of back and forth between diploid and haploid. Terminology that's semi-important to know. When we're in the diploid phase, so down here in the red, kind of the terminology associated with that is sporophyte, and then in the upper half, haploid cells are known as gametophytes. Whoops. And again, it says it down here, but plants do this haplodiploidic style, and some algae. So algae are kind of, they can do one or the other, not both. Okay, hopefully that makes sense. Again, this is something I also find kind of confusing, just from in my opinion, and from personal experience. This is something I had a hard time wrapping my head around the first time I learned this. So again, I don't think it's something that most people can it just clicks the first time you see it. And so it might be helpful to kind of go over it a few more times if you're still a little fuzzy. And of course, at any time you can ask me any specific questions that you have about it. Okay. So why do these different patterns exist, these different cycles? Why is there variation in how sexual reproduction occurs? Well, probably because there are advantages to each, which makes sense. So each of these are kind of suited for a different scenario. So what are the advantages of each of these three life history patterns? Well, diplontic is advantageous in unstable environments. And why this is diplontic, or organisms that use this diplontic cycle are diploid almost all the time, with the exception of gametes. So they have 2 times the chromosomes, which means they'll have 2 times the mutations, just purely based on the fact that, you know, it's if you have, I won't get into it. I think that's pretty clear. You double the chromosomes, you're going to get double the mutations. If you have the chromosomes, you're going to have half the mutations. So you're going to have more mutations in diplontic life cycles because the cells are diploid. And mutations are generally a good thing in unstable environments, environments that are changing. Pretty much any natural environment. Lab conditions are pretty much the only place you can get a really stable environment. Just think about, I mean, global warming, climate change is acting on nature currently on a macroscopic scale, but there's always going to be small changes in the environment as well. Haplontic, that's going to be, you could probably guess, because haplontic and diplontic are kind of opposites of each other, but this is going to be advantageous in stable environments. And that's because you have half the chromosomes, you're going to have half the mutations. You're good to go if things aren't changing. And again, like I said, why fix what isn't broke, or don't fix what isn't broke, I guess. If an organism is surviving in an environment, the environment's not changing, why change the DNA of the organism? Because it's working out, so, you know, you don't really want mutations to come in and change it. So haplodiplontic, it's like Hannah Montana in a sense. You're getting the best of both worlds. Because alternation of generations means that you can exist as either diploid or haploid depending on what situation is going to be best for whatever environment that they're in. Different stages for different environments, I guess we'll say that, just to summarize. So yeah, literally, if this haplontic, this haploid lifestyle isn't working out, well, the next generation will be diploid. That'll probably work out better. Okay, so that is all I had prepared for this evening. I know it's kind of a lot. It was only 2 lectures, but I find these 2 lectures are kind of heavy, personally. But if you have any questions whatsoever, feel free to let me know. Otherwise, you're free to go. Have a good week. It's Monday. I keep thinking it's like Thursday and I keep getting disappointed, but